about bacteria and how infections become resistant to bacteria, and they do. So if your doctor gives you an antibiotic to treat something, and you start to feel better, that is not the sign to stop taking the antibiotic. That means that, yes, it's working, but you need to keep taking it. Because those last few pills that you take are what's going to kill those really tough bacteria. It's like a gang. You're in the black yoga pants gang, and you take out the first couple of members because they're weak. They're weak. You took them out easily. Then the next thing you know, the biggest, strongest ones are the last ones to go down. So you have to take all of the antibiotic to take them out. And that's genetic. So before you take the antibiotic, you have all those different levels of resistance of bacteria. You start taking it, you start knocking out those weak guys, and then you get all these strong guys. So you take the last of it and you knock them all out. If you don't, then you've got all the tough guys sticking around, and then you have to take a stronger antibiotic to get rid of them. So that was just a reminder about taking your pills. So bacteria were interest to us because women in particular, they are more vulnerable to urinary tract infections than men. So we wanted to study a little bit about it.
All right, so our procedure initially focused on taking two commonly found strains of bacteria found in urinary tract infections, which were Klebsiella pneumoniae and Proteus mirabilis, and doing bacterial lawns on petri dishes and using the Kirby-Bauer test to see if baking soda or unsweetened cranberry juice would have an effect on bacterial growth by measuring zones of inhibition.
Oh, I'm sorry. So we should be. So we wanted to do direct treatments on these specific bacteria because these particular bacteria have not specifically been studied, and we wanted to because although E. coli is the most commonly found implicated bacteria in UTIs, these are the second most.
And we couldn't get our hands on the strain of E. coli because it requires like a biohazard 5 level clearance.
We don't have any clearances. So we use these guys, Klebsiella pneumoniae.
All right, so both of these are opportunistic human pathogens, and they're both found in septicemia, which is infection of the bloodstream, urinary tract infections, and then Klebsiella is specific to pneumonia, meningitis, diarrhea, and soft tissue infections.
And that's an electron microscope photograph of Klebsiella. You can see it's rod-shaped there, up close and personal. And then the other one that we studied was Proteus, which a lot of folks in the class used. Also gram-negative, also rod-shaped, but this one's a little bit different. This one can exhibit what's called swarming motility. So although it can form colonies, it can also swarm and spread out. And that kind of looks cool, like that. I don't know if how many orgo people did the lab where we used IR spec and then they kind of, that was pretty cool. So it kind of looks like that.
All right, and these bacteria are also involved with blood infections, pneumonia, and wound infections, mostly in people in the hospital who are being treated post-surgery or some sort of injury that they've come in for treatment on.
And that's what Proteus looks like on a specific type of agar plate. They like to use the blood agar plates for these. It's kind of cool, but kind of weird. So materials and methods. What do you always have to have in lab? Indeed. That is Brandon in the back. He was eating something, I don't know what it was. So look at all that.
Yeah.
There's our bacteria.
And this is all for the initial procedure that we were going to use with the slant cultures. What we found was that after we did our slant cultures, left them incubate at 37 degrees Celsius, we had no growth. And we contribute that most likely to the fact that when you're doing the aseptic technique and you put the inoculating loop in the flame, you can't measure the temperature of that inoculating loop. So we probably stuck it in the bacteria too hot, so we killed the bacteria before we were even able to get it in the cultures.
We tried to be patient. We tried to take our time, but I think we killed them.
So when we revamped the procedure, what we ended up doing was using the straight broth cultures of the bacteria, looking for a concentration of 0.5 or up to make sure it would grow fast enough, there was enough growth for us to test treatment.
So just like most people who worked with the bacteria, we all did the same procedures. We did the autoclaving, we created the broth, we made the agar, we tested the optical density, created some plates, which is Alex so kindly mentioned. Nothing happened there, but we tried. So then, we tried to create something really cool. I've never heard of slant cultures before. These are neat. Has anybody here made them? Is anyone familiar with them? So you put the agar in the test tube, and then you tilt it, so then you increase the surface area, and you could do your cultures that way. You could.
But we didn't.
Yeah. It's kind of sad because guess what happened there? So try number 2. It wasn't good. So we created broth cultures. That was good. I really liked the idea of broth cultures because to me it was closer to what we would experience in the human body. You didn't have to do anything special to it. The bacteria is there, you apply the treatment, and see what happens.
And the incubation was also easier because 37 degrees Celsius is around average human body temperature in Celsius, so it made it even more accurate.
So we prepared some treatments. This was the most difficult one to prepare. We measured out 1 gram of baking soda. We added enough distilled water, which we know isn't sterile now. Doesn't matter though. We put it into graduated cylinders, put our thumb over it, then shook it like crazy, shaken, not stirred. And we added it to our broth cultures.
And we actually did have some red cultures on purpose.
As some people.
A couple of the other fruits.
Because we added 5 milliliters of no sugar added cranberry juice directly to our broth cultures. And then we incubated them in what looks like a refrigerator, but it's a lot warmer and really stinks. We had some results with the broth cultures, which was pretty exciting. So like we said, we tested the 2 home remedies that are pretty common. If you go onto websites, you Google home remedies for UTIs, you're going to find these along with some of the others. Like we also mentioned, usually E. coli is the most mentioned bacteria. We tried against the second 2 most common, Klebsiella and Proteus. And we hypothesized
that if these were both effective treatments, we would see a constant decrease in the optical density of the treated bacteria, and the control will continue to increase.
And we kind of did see that. We did see the controls slightly increase.
Yes.
And we consistently saw with the baking soda treatments
The decrease, and then the cranberry juice, actually, within both of our trials, we ended up with contradictory results. In trial 1, the optical density increased when cranberry juice was added, but in trial 2, it decreased.
So the only difference we had was the initial optical density in trial 1 was lower than in trial 2. I don't know if that has anything to do with it or not. I mean, we would have to run the trial again to see if starting with a higher concentration, if the cranberry juice is more effective because there's more, I don't know. But we talked about the fact that we were applying this no sugar added cranberry juice directly to the bacteria. It's not how it happens in the body because it has to go through your mouth and the rest of your digestive tract. So we thought if we did this again, we would subject it to amylase, which is one of the enzymes in your mouth that breaks down starches, which we would have found in no sugar added, because even though it's no sugar added, there's natural sugars.
Yeah.
And hydrochloric acid, because you have that in your tummy, and that helps break down some things too. So that's what our baking soda treatments look like. So some numbers. Let me zoom in on this a So as you can see, our controls would go up slightly, but then the baking soda numbers dropped like a rock, but the cranberry juice numbers climbed slightly. That was trial on day 1. Trial on day 2. Again, you see the control readings go up slightly from the initial reading, then the baking soda numbers nicely dropped. Cranberry juice rose.
And what was interesting was that the cranberry juice was increasing the growth rate of the optical density faster than just the control in the incubation, which is kind of worrisome.
Yeah.
Because if this is how it actually occurs in the body, then, everybody's saying use this treatment, but it's actually just going to make your condition worse.
And again, an hour later, we see baking soda still decreasing, and we see cranberry juice still increasing. But then, like we said, when we looked at, when we did the second trial, we looked at the numbers, not sure what's going on here with this control reading. It jumped like crazy. Don't know why. This one jumped quite a bit too. Baking soda dropped like crazy on the second trial, dropped for Proteus, but then the numbers decreased for the no sugar added cranberry juice. I'm not sure why. Like I said, we could hypothesize that it was because we had different starting numbers.
And another common factor in a lot of the research I was reading was that cranberry juice doesn't actually have an effect on the optical density of the bacteria themselves, but it's an irritant to the urinary tract. And when it inflames the urinary tract, it somehow makes it harder for certain types of bacteria to attach to the wall, which would obviously not be displayed in here because we're not running it through a urinary tract. We're not running it through anyone's system.
Right, we're just applying it directly to bacteria. And there's a famous report, the Cochrane report, that's looked at thousands of studies. And what they found was taking the supplements, the cranberry juice supplements in pill form, is one of the few things that they could definitively say, yes, this does reduce the incidence and recurrence of urinary tract infections in women. And they studied it over a 12-month period. In our study, we have conflicting results, but again, we didn't do anything with the digestive tract, so that might be why. And if that wasn't enough for you, there's some graphs that say exactly what we just said, but only prettier. So, what? We know that UTIs are a common bacterial infection in women, and a lot of women seek home treatments. I mean, part of the reason is insurance. A lot of women don't have insurance, or it's expensive to go to the doctors, or maybe antibiotics make the time. So what?
We wanted to be able to answer this question and see if these home remedies actually did have an impact on the bacteria. And we were able to conclude in terms of our hypothesis that our hypothesis was in part correct, but in part not, because antibiotics will wipe out the bacteria entirely. You will have an optical density of 0. And while we didn't run the trials long enough to get that low, we saw the baking soda continue to decrease over the time. So if we would have had more time, we could have run the trials longer to see if we would have reached that optical density level.
Now what? Sorry about that, Professor. All right, so the baking soda results, they're promising, but we do need further testing. And based on what we've had, I would honestly try the baking soda sitz bath if I had a UTI at home. I think we would have to test the cranberry juice, like we said, with the amylase and the HCl, or even just the pill, something different.
What also has to be taken into consideration here is urinary tract infections. If you don't decrease that bacterial growth fast enough and you don't kill off the population fast enough, it's going to spread up to your kidneys, it's going to get into your bloodstream. So these are not really, it's not necessarily an easy thing to research because these people need immediate treatment or else, they can come out with some pretty life-crippling results.
That's true. That is it.
Any questions?
Oh, lots of questions. I'm scared. Yes, Chad.
So I know with like things like yogurt, the idea is that the bacteria in the yogurt can kind of like make it more competitive for the bacteria that's causing the yeast infection. But do you know the mechanism for like how the cranberry juice works?
Like Alex was pointing out, they think that it's an irritant to the bladder actually, so that it won't stick to the walls and let them grow. So I mean, we wanted to study yeast infections, but the things that we needed, the clearances that we needed to be able to get the yeast to do that, we don't have the biohazard 4 level that we would need. But it's an interesting question. So no, I don't know the exact mechanism. Thanks for asking me such a hard question.
Sam?
You said your cultures weren't viable, but your science cultures weren't viable. Like, what do you think led to that? Finality?
Inaccurate temperature of the inoculating mold. We ran it through the flame, waited till it got hot, let it cool. Probably didn't let it cool enough because there's no accurate way to get a temperature reading on that.
Okay.
So there may have been 1 or 2 sizzles in there.
When she says 1 or 2, she means all of them. We killed them all. It was an accident. Yes.
Why exactly was growth not measured, and why was only optimal density measured of the bacteria?
At that,
We were working with broth cultures at that point.
and that means they were simply in the test tubes. We tried plating them, and nothing grew. We tried doing slant cultures, and none of the bacteria grew. So we wanted to get some results, so we went to broth cultures, and we were happy with that because, like I said, I think it more accurately imitated what you would experience in your body than a plate.
Did you give it time, like after you treated the bacteria with like the baking soda or the cranberry juice? Did you wait like a 20-minute period? Did you like just take the occupancy right away? Like how did you do
For
that?
trial 1, there was a 1-hour period for the first treatment, and then we took a second reading after 24 hours, and then a third reading at 25 and a half. Trial 2, we only got 1 reading after 6 days to give it a longer span.
Professor.
So, when people use baking soda as a treatment, so I'm assuming that are they mixing it into?
You mix it
a
with
warm-
warm water and you sit in it. It's
And it
called a
sits
sit
back.
bath.
Okay, all right. Okay, because I was wondering.
How do you get it in there?
Well, if you drink or consume baking soda, that changes the pH in the stomach, which can be a bigger problem than trying to get it to the bladder.
Yeah.
Yeah, we wouldn't recommend eating.
Yeah. Don't
baking
please